Pure cultures of bacteria are most often obtained by the streak plate technique. In this procedure, an inoculum of bacteria in nutrient broth is placed on a solid nutrient agar and then streaked to separate individual cells. To produce a successful streak plate, you will use aseptic technique to prevent contamination of your plate by environmental microbes. You will inoculate the agar using a 4-streak technique as shown. Use a permanent marker to label the bottom of the plate with your initials, the date, the type of medium, and the culture or source of the inoculum. Labeling the bottom of your plate ensures that the written information stays with your culture, even if the lid gets accidentally exchanged with another plate. We will use a loopful of Staphylococcus aureus culture to inoculate the plate. Hold the loop like a pencil. Flame the loop until it is red hot, and then let it cool. Remove the cap from the tube and pass the mouth of the tube through the flame. Obtain a loop of bacteria from the tube and then pass the mouth of the tube through the flame again before recapping. Return the broth tube to the test tube rack. You won't use the tube further in this procedure. Lift one side of the Petri plate lid and streak the first quadrant of the agar with as many tightly spaced back and forth streaks as possible. Take care to keep the loop in contact with the agar, but do not gouge the agar. Replace the lid, flame the loop, and let it cool. In the next steps, you will spread some of the bacteria from the initial inoculation into other quadrants of the Petri plate. You only want a tiny fraction of the bacteria in one quadrant to be spread to the next, so it's important to flame away all bacteria from the loop in between each streaking step. Using the sterilized loop, streak a few times across your first streak and into the second quadrant. Then, with the loop still in contact with the agar, streak several more times within the second quadrant. Replace the lid and rotate the plate. Flame the loop and let it cool. Using your sterilized loop, streak a few times across your second streak and into the third quadrant. Then streak several more times within the third quadrant. Replace the lid and rotate the plate. Flame the loop and let it cool. Using your sterilized loop, streak a few times across your third streak and into the fourth quadrant. Then streak several more times within the fourth quadrant. As you streak into the center of the plate, take care to avoid touching any of the streaks in quadrant 1. Replace the lid, flame the loop, and let it cool. Follow your instructor's directions for disposal or further incubation of your original broth tube. Incubate the plate in an inverted position at 35 to 37 degrees Celsius for at least 24 hours. During incubation, water evaporates from the agar and will condense when it reaches the cooler Petri plate lid. If the plate is properly inverted, this condensation will stay in the lid, away from the agar. If the plate is improperly incubated with the lid on top, the condensation will rain back onto the agar. Bacteria will move in the water, and you won't see discrete colonies. Note that the lid is removed from this plate to clearly show the effects of improper incubation. Do not remove the lids from your own plates. Discrete colonies are visible in this correctly incubated plate. Each colony is derived from a single cell or small group of cells that has undergone many rounds of cell division. Bacterial species produce their own distinctive-looking colonies with regard to colony shape, color, and size. This plate was inoculated with a pure culture. All the colonies are identical. This plate was inoculated with a mixed culture of 2 different bacterial species. One species produces red colonies, and the other makes smaller yellow colonies. After recording all of your data, follow your instructor's directions for disposal or further incubation of your plates.